the PhilInBioMed Award is reserved, as the name says, for those pincers, philosophers in science, that do philosophy in biology or medicine. So this is very simple to understand in principle, but it elicited a lot of misunderstanding. So for that reason, it is important probably to remind you clearly what philosophy in science is not. So indeed, most of the time, philosophers engage into the solution of philosophical problems. And most of the time, the solution to philosophical problems is completely orthogonal to the solution of scientific problems. Sometimes, though, philosophical problems happen to also be scientific. That is, a philosophical solution to a philosophical problem may unlock as well a scientific problem, for instance, by dissolving some conceptual confusion. Most philosophers of science probably would welcome the view that some of their work might contribute to science as a side effect, while at the same time they are perfectly happy to consider that this is absolutely not required. Maybe a small number of them would only, a small number of them only would look exclusively for the solution to philosophical problems that have no possible impact on science itself. So PINS is very different from that. And to understand why, it is required to emphasize the consequences of a radical shift from philosophy as defined by philosophical problems to philosophy as defined by philosophical tools. A philosophical tool can be a certain skill, particularly developed by philosophers, for instance, conceptual analysis, or a particular sensitivity to the clarification of arguments, or certain concepts or conceptual distinctions as they are proposed in the philosophical literature. So to a pincer, using philosophical tools is enough to define a work as philosophical, while it is not necessary that the tool itself is applied to a philosophical problem. But to many philosophers, on the other hand, not using these tools to solve a problem that is to some extent philosophical is sufficient for this work not to be philosophical anymore. So this is a main reason why a strong emphasis on philosophy in science may often be rejected from philosophy of science journals or conferences with the judgment that this is not philosophical, it is scientific. So indeed, contemporary philosophy of science is not afraid of scientific technicalities, but it is uncomfortable when the philosophical question is out of sight. And I insist, of course, on the idea that the philosophical question is out of sight, not the philosophical tool. A second reason is that many philosophers would fear or even loathe any violation of the Lockean division of labor between scientists and philosophers. They would, for very good reasons, recall the series of calamitous injunctions philosophers of science have often thought they were entitled to place on scientists. So in contrast, the goal of the PhilInBioMed Award is to encourage philosophers not simply to engage into more scientific details, but to raise and contribute to the solution of scientific problems. So this is not to propose that philosophers have dual careers, neatly separated as philosophers and as scientists, but that they should contribute qua philosophers. Indeed, philosophers do not necessarily do badly in science in general, simply because they are philosophers, but they simply just did badly maybe this time or that time, or maybe most of the time, but not necessarily in principle and all of the time. So it will never be emphasized enough how hard it is to harness philosophical skills and concepts to scientific relevance, and in the sense of contributing to the solution of a scientific problem. So it is already very hard to use it in a way that is relevant for science, that is to make a scientific proposal, but it is even harder that this proposal becomes a significant contribution to science. So PINS is very hard, and when it is successful, it is useful. And this is a very good reason to encourage it. And this is exactly that quest that the PhilInBioMed Award is promoting. So thank you for your attention. Thomas, if you want to say now a word for our first awardee.
Thank you very much, Maël. I really want to insist again on what Maël just said. Basically, thank you, Elliott. Thank you, Silvia, Kevin, and Ford for accepting to be here today or remotely here for Ford. And what I'm going to do now is to explain in just a few words why Elliott got the prize. I mean, maybe this is obvious, maybe not. In case you wonder, this is Elliott yesterday at the Père Non-Père Cave near Bordeaux, where we had a great time. If we have a look at the bibliography of Elliott, I think we all know it's a very impressive bibliography. 14 books, in fact, 15 books. One is forthcoming. Several of them have several editions. I think it's fair to say that the book that has had the most influence, I think, on biology is The Nature of Selection, 1984. But it's also probably the book that had the strongest influence on philosophy of biology. Elliott published no less than 293 papers since 1976. That's a lot. If we move to a little bit of qualitative analysis to explain why Elliott got this prize, I think it's very important to start by what Maël just explained, with it, which is that if you want to do philosophy in science, the judgments of scientists is absolutely central. We are very much dependent on the extent to which scientists will consider what we do as philosophers as important and impactful. And this is exactly something which is very striking in the work of Elliott. People very often doing biology themselves said that the best possible description of Elliott was that he was clearly a philosopher, but a philosopher who makes a difference to biology. Elliott published his book in 1984, The Nature of Selection. In 1986, Ernst Mayr, who, as you know, is extremely famous and was very influential at Harvard, wrote a book review in 1986 in Paleobiology. And I think that book review was instrumental in making Elliott visible to the scientific community. Not that he needed to be visible, but suddenly you become incredibly visible because someone like Ernst Mayr says something like that. Sober has given us what is perhaps the most careful and penetrating analysis of the concept of natural selection as it affects the process of evolution. Working my way through Sober's book made me realize the ambiguity of much of the evolutionary terminology. Both biologists and philosophers will benefit greatly from the study of Sober's careful analysis of the problems of natural selection. Dick Lewontin, who was probably the most important population geneticist of the 20th century, also wrote very nice things about this book and about Elliott about the same time. Another view, which is also very interesting, is that Elliott is so much good in theoretical biology, he's so good in theoretical biology that he's not a philosopher anymore. This is a very classic view. When you're accepted by scientists as a scientist, you cease to be a philosopher. And for example, Ford, who is one of the commentators today, explicitly said in an interview, the philosophical questions are useful. And certainly philosophers like Elliott Sober, who is basically a theoretical biologist as far as I can tell, are very useful too. What are the key ideas for which Elliott Sober is known? I think it's just a small list, but I think those ideas are very important. Selection of, selection for, this distinction is important to emphasize, for example, the fact that many things that could seem to be selected are in fact byproducts of something else. How adaptation and adaptationism should be defined. 3, Elliott endorsed a certain version of group selection, and he did that particularly in collaboration with David Else Wilson in Reviving the Superorganism, 1989, and in this book and 2 others that was published in 1998. Another idea is cladistic parsimony, where Elliott intervened into a debate between cladists and likelihoodists, and this is something that I think will be related to some of the topics that will be discussed today. Elliott was also involved in discussions over how you build model in science, and I think it's fair to say that a lot of what Elliott has done about this is a long response to the very influential paper by Richard Levins on model building, and this is work that Elliott has done in collaboration with Steve Orszag in particular. In this very nice paper published last year in Philosophy of Science, Philosophy in Science, Some Personal Reflections, Elliott gives this advice to the young people who are interested in doing philosophy in science. If you want to do philosophy in science, you should get interested in controversies, in where scientists disagree. And when we have a look at what Elliott did during his great career, this is quite a lot about that, identifying places where scientists disagree and intervening in those controversies with something which is going to be a positive contribution to that debate or that question. If we go to more quantitative quantitative analysis, which means mainly doing a little bit of bibliometrics, where the main idea will be to try to determine to what extent scientists and biologists in particular pay attention to the work of Elliott and sometimes Elliott and co-authors. The first message, which I think is super important, is that even Elliott, as famous as he might be among biologists, has his first community history and philosophy of science. So if you have a look at the citations to Elliott's work, it's first and foremost historians and philosophers of science who cite his work. Maybe this is not surprising, but this is something important to emphasize. If we now move to another question, which is, which are the biological domains that cite Elliott the most? You will not be too surprised. So this is all based on the Web of Science. Evolution in biology comes first, then ecology, genetics, heredity, general biology, zoology, more surprising, biochemistry and molecular biology. If you are interested in who are the biologists who cite Elliott the most, you will find very familiar names if you know the topics on which Elliott has worked. That includes co-authors, for example, of Elliott, including David Wilson or Steve Orzac, for example, but also people we know very well in France, Guillaume Lecointre, who is in the Muséum d'Histoire Naturelle in Paris. And the one who has cited Elliott the most is Arnold Klug, who is a very famous cladist who was the president of the Society of Cladistics and in charge of the journal for years. Now I'm taking one example just to illustrate the kind of work that Elliott has done. This is not just an example taken by chance or just as another example. This is a very, I think, important example because this is the most cited paper by Elliott, by scientists, by biologists, if you have a narrow concept of biology, which means that you exclude psychology. If you include psychology, then the group selection papers, one group selection paper becomes more cited than this one. This is just a question of bibliometrics, but I think it's interesting to understand what's going on in that paper. So basically, that paper is a re-examination of the long-standing debate in evolution biology about the possibility of natural selection acting at the level of groups and or the level of superorganisms. And as you know, one example of superorganism would be an insect colony like termite colonies, for example. And here, I think this is a very good illustration of PINS as a conceptual clarification and perhaps even more importantly, as checking logical consistency. The paper is quite a lot about that. The paper proposes that superorganisms, which are defined in the paper as collections of single creatures in which between unit selection overwhelms within unit selection, are not only logically possible, but they really exist in nature. And the paper is very interesting because it gives very interesting different examples of things that count as superorganisms. And it also says all that does not count as superorganisms. So these are not superorganisms, these are superorganisms, given that definition, which I think is very operational and very interesting. It exposes the logical inconsistency of people who would exclude a priori superorganism. The paper says if you exclude a priori superorganism, you would have to exclude many definitions or many understandings of organisms as well. And this is probably not something you're ready to do. So I think this is a very powerful paper. It has been cited regularly over the years with some peaks, certain years. I don't know exactly why. And also, you probably don't see the details, but these are the most cited papers in biology that cite the paper Reviving the Superorganism. So these are very influential papers that happen to cite that paper. Some of them will talk a lot about the paper. Some of them will just mention the paper in passing. And of course, that makes a difference. Very interestingly, if you could zoom in here, you would recognize some names. For example, Chasmary and Maynard Smith or Colin Strassman, Stephen Frank, and many people who we know do theoretical biology in general. It's interesting to notice that this paper in general has been widely used and discussed in the scientific literature, particularly by biologists interested in testing whether this definition of a superorganism, the one defended by Wilson and Sober, works for a particular species. This is the case in that paper, for example. And I think even more importantly, some people like Maynard Smith and Chasmary really used that paper to make a novel scientific proposal. And I think this is one of the best things that can happen to a philosopher when your idea is taken up and used to propose something new in a given science. A more recent example is this, in this paper by Theo Beld in Nature in 2010. it's very interesting to see, well, you see the title of the paper, a single author in Nature is very rare. So it's a very special paper, but it's also a very interesting theoretical biology paper. And you can see here that the first person cited is Elliott. And here again, Sober and Steele are cited for their papers in the Journal of Theoretical Biology. So I think it's fair to say that Elliott is not only one of the most important philosophers of biology alive, he's also one of the very few philosophers of sciences who have had a strong impact on the science. And for that reason, I think it's perfectly fair that Elliott receives the PhilInBioMed Award. So congratulations, Elliott. And now we will go to the very interesting stuff, which is the lecture by Elliott, then the commentaries, and then the replies by Elliott. Elliott, the floor is yours.
Okay. Well, thank you very much for this great honor. I'm rather embarrassed and nervous about this whole thing. I should say for maybe it's worth me saying that I didn't plan my career such that I would be called an advocate of PINS. I just by accident stumbled into scientific controversies that turned out to have a philosophical dimension as I saw it. And I don't think it was like I was the only one who noticed this. I mean, the scientists themselves realized in group selection controversy and controversy about phylogenetic inference that these were questions that you can't just solve by getting a data set and the observations tell you. And they would fight with each other doing philosophy. When you see scientists as a last resort being philosophers, you know, you as a philosopher think, hey, I could maybe do that too. So it was, I mean, so many of us in my generation of philosophers of science sort of saw this going on, and we were attracted to these controversies just because they had philosophical content, and we thought this would be fun and maybe we can help. So thanks again. Do I have a pointer on this device? So this button here, the top one?
The red one.
Red one. Right. Okay, good. So I'm going to talk about two areas. Actually, I just mentioned the two areas that I'm going to talk about. The first one is about group selection. And I'm going to talk pretty much solely about Darwin's views about this, and then bring in an idea from 20th century statistics, which I think crystallizes one of the conceptual difficulties that I think most of us, me included, had in thinking about how altruism could evolve, this thing called Simpson's paradox. And then the second thing about phylogenetic inference, I'll talk about Darwin's views connected with that. And then I'll talk about this tool, again from 20th century statistics, called the law of likelihood, and how that allows you to clarify and refine some of Darwin's cool ideas about what counts as good evidence for common ancestry and some other things too. So when I was just getting into this group selection thing decades ago, many biologists and historians and philosophers who thought group selection was not just factually mistaken, but a stupid, confused, waste of time kind of idea, they cited Darwin as their pal, that he had his head screwed on right. He rejected the idea of group selection. And, you know, if you can show you're doing what the genius Darwin did, you know, you're in good shape. It turns out it doesn't take a lot of digging to see that Darwin thought that group selection was a real and important cause of evolution. So here I'll talk about three of the examples from Darwin. So that's a blow up of the barbed, whoops. How did I get all the way back there? The barbed stinger, which Darwin calls backward serratures, that a honeybee's stinger has. And Darwin says, can we consider the sting of the wasp or the bee perfect, which when used against many attacking animals cannot be withdrawn owing to the backward serratures, the barbs, and so inevitably cause the death of the insect by tearing out its viscera. And then he goes on to say, Well, actually, if you think about it, a nest of honeybees that contains organisms that have barbed stingers is going to do better in getting predators, intruders to the nest, like a hungry bear, to get out of there and not stick its nose in, because this barbed stinger gets stuck in the bear, even though when the bee pulls away, it kills itself. The barbed stinger continues to pump venom after the bee is dead. And Darwin's reasoning is that nests that have organisms in it that do that basically are suicidal in terms of the way they repel hungry bears, will do better, he theorized, than nests in which all the stingers are not barbed. So that's why the barbed stinger evolves. It's not good for the organism that has it, rather it's good for the nest in which those organisms live. And so in the competition of groups against groups, group selection, you have this trait evolving because it provides a group adaptation. Social insects, Darwin says in The Origin that the idea that there are sterile individuals in many species of social insect was for him one of the giant problems that he himself wrestled with. Not early in the process of writing up his theory, but rather much later. And he basically says, in posing the problem, I mean, sterility is not good for the fitness of an organism. It means you have 0 babies. What could be worse than that? The reason he theorized that sterility evolved as a characteristic of some of the members of a nest in social insects is that these sterile workers are not just sitting around being sterile, they're helping build a nest, take care of young, they're doing things that actually benefit the community. And so communities that contain sterile workers will do better than nests that contain only fertile workers. Again, group selection is the explanation why a trait that is deleterious for individuals actually increases in frequency in evolution. Answer, it's good for the group, though it's bad for the individual. And then the third example might be the most famous one. It's not in The Origin. It's in a book that Darwin wrote about human evolution, The Descent of Man. And he's puzzled as to why in times of warfare, brave men come to the front. He's talking about men here. And they risk their lives, sometimes sacrificing their lives to defend the group. I mean, and he says that's not good for the survival and reproduction of the individuals who do this, these courageous men, but it's good for the group. And when groups compete with groups, as Darwin thought the whole history of humanity has been like, it's been a warfare, group against group, groups in which individuals are prepared to sacrifice their lives or their own welfare for the sake of the group, win competitions of group against group. It's good. Groups that are made of good cooperators outperform groups that don't have good cooperators, where cooperation sometimes involves sacrificing your own life for the sake of the group. So those are 3 of the examples. That's Darwin's solution to it. Now some terminology that's now current, but Darwin didn't use these words. Evolutionary biologists now talk about a characteristic, an altruist is somebody or some organism that confers a fitness benefit on another individual at a fitness cost to itself. And by fitness, I mean the ability to survive and reproduce. So you're increasing so think of the sterile workers having reduced fitness in terms of their own reproduction, but enhancing the reproductive success of other individuals in the community itself. And selfish just means that the individual does not give away fitness benefits to others. It has nothing to do with these terms have nothing to do with psychology in contemporary biology. Mindless organisms can be altruistic or selfishness just in terms of how their traits affect their own fitness and the fitness of other organisms. And even organisms like us who have minds, the definition of what it would be for a human being to be altruistic has nothing to do, in the first instance, with psychological motivation and everything to do with the effect on others and on self in terms of fitness. Okay. This is the kind of diagram that I'll be using in several occasions today. It's called a fitness function. There are different possible groups that contain a certain percentage of selfish individuals. And you'll notice in this diagram that no matter how common or rare selfishness is in a group, selfish individuals are always fitter than altruists. So if you're in a 50% group, that's the fitness of the average altruist, that's the fitness of the average selfish individual. And this is kind of true by definition of what selfishness means in this terminology. And the other thing to notice is that the average fitness of the individuals in a group goes up as altruism increases in the group. So over here you have a group that's near 100 or nearly 100% altruistic. The average fitness is somewhere over here. This is a group that's 99% selfish, and the average fitness is over here. So I didn't draw this line, but I should have drawn a red line that goes from here, straight line to there, which represents the average fitness of individuals in a group, sometimes called W bar, the average. So those are the two properties, and it's just a way of saying, of representing the idea that selfishness is good for the individual as compared to the other individuals within the group. And secondly, groups do better the more altruists they contain. That's what this picture says. And here's an argument that I think, I know I stumbled over this a lot when I was starting to think about this, and I think it's a kind of hurdle that everybody has to come to grips with, and just thinking about how altruism could ever evolve by natural selection. Here's the argument to consider. So the first premise is that by definition, altruists are less fit than selfish individuals. It seems like, didn't I just say that? That seems the definition of altruism and selfishness. Second premise, when natural selection controls the evolutionary process, fitter traits increase in frequency and less fit traits decline in frequency. So if you have fast and slow zebras in a herd and the fast ones evade lion attack more successfully than the slow ones, if natural selection is going to control this process, what you expect is that fast will increase in frequency, slow will decline. So what seems to follow from this, these two premises, which seem clearly right, is that altruism cannot evolve by natural selection. Rather, what should happen is selfishness is what's going to evolve. By evolve, I just mean increase in frequency. And if altruism can't evolve by natural selection, it can't evolve by group selection either. Group selection is just a kind of natural selection. So it looks like Darwin goofed. I mean, the whole idea that group selection was the solution to the problem of why altruism exists in nature seems wrong from the start. How embarrassing for the great genius. Well, I think this argument is flawed, and now I want to pick it apart. So I think I may have deleted something here. Anyhow, so no, that's wrong. First of all, what's wrong with this first premise is that what's true is that if altruists and selfish individuals live in the same group, by definition, the selfish individuals are going to be fitter than the altruists. That's what's correct. But when you look at the entire ensemble of different groups, it doesn't follow from that the average altruist is less fit than the average selfish individual. So there's something wrong with this part of the argument. That part is okay. And what we should have down here, I apologize that some slides are missing here. I don't know how this happened. What's true is that altruism can't evolve by selection if selection means selection within a single group. You have one group and you have altruist and selfish individuals in it, natural selection will lead selfishness to 100%, altruism to 0. But if you have a bunch of different groups, the whole story changes. And now I'm going to turn to Simpson's paradox. I just want to look ahead here. Oh, the slides are out of order. So what's wrong with this? I'm going to just do it as I hope to do it. There's an ambiguity in the first premise. When you say by definition altruists are less fit than selfish individuals, does that mean in the same group, or do you mean in a two-group meta-population? Actually, I'll go back and do it this way. Again, I apologize. Okay, Simpson's paradox. So don't forget, but put aside the stuff I was just saying about altruism and selfishness. This is a completely different subject. It's not a hypothetical example. In the 1970s, it was observed at the University of California at Berkeley that women were to graduate school a lot less frequently than men were. And of course, what was immediately suggested by that finding is that departments were discriminating against women. But when the administrators looked at the admission and rejection rates within departments, they discovered that men and women were getting into grad school with equal frequency. And how can these two observations both be true? And once I get through with an example of this, I hope you'll see how that's possible. Okay, so I'm going to make up an example now. This is a university that has two departments, D1 and D2. And this is there are 200 people who apply. 90 out of 100 males, 90 of them apply to Department D1, and 90 of the 100 women apply to Department D2. Here are the admission rates. It's harder to get into Department D2. They only take 20% of the applicants. Easier to get into Department D1. And so what happens when you figure out how many individuals got in here? You take 10 and take 40% of that, you get 4. You take 90 individuals here, 20% of them get in, so they're 18. So notice that because the departments differ in their admission rates, although the departments are the same, each department treats men and women the same, it turns out that out of the 100 women who apply, 22 get admitted, and of the 100 men, 38 get admitted. So within each department, men and women have the same chance of getting in, but when you average over these two different departments, with men mainly applying to the easier to get into department, and women mainly applying to the harder to get into department, you find that when you average across these two categories, women get in less often overall. Now I want to apply that idea, just to take the same idea and apply it now to group selection. And so there's the picture I had up there before to just talk about the definition of altruism and selfishness. And I want to talk about two different groups. Group 1 is 10% selfish, 90% altruistic, and group 2 is 90% selfish. And those are the fitnesses of selfish and altruistic individuals in group 1, selfish and altruistic individuals in group 2. So let's see what happens when we start with two groups, each containing 100 individuals. The first group is mainly altruistic. The second group is mainly selfish. Here are the fitnesses of individuals. Notice that groups in which there are lots of 90% altruists, everybody's fitter than the individuals who live in groups that are 90% selfish. And so when you have those numbers, what you get is just like what happened at Berkeley. Notice that 3 times 90 means 270 individuals. This is the next generation. This is the fitness means the number of babies you have if you are an altruist living in group 1, a selfish individual living in group 2. So you just multiply these two tables together. 280 altruists in the second generation and 220 selfish individuals in the second generation. And so what's happened here, you started with altruism and selfishness in the first generation being 50%, and now altruism has increased in frequency. So now let's go back to that argument that seems like a stumper, like how can this make sense to talk about group selection increasing the frequency of altruism? It looks like by definition you can't, that's what that argument says. So there's an ambiguity in the first premise. When we talk about, by definition, altruists are less fit than selfish individuals, are we talking about altruistic and selfish individuals in the same group? Or are we talking about altruism and selfish individuals looking across all the groups in this ensemble? And in fact, the definition is about selfish and altruistic individuals in the same group. The conclusion talks about natural selection. But what we're actually talking about here, if the conclusion is going to be true, is that altruism cannot evolve by within group selection. And now we have a valid argument. This is the true part of what's present in this flawed argument I started with, that if you've got only a single group and altruists compete with selfish individuals, altruism is going to go to 0. But that says nothing about whether group selection can cause altruism to evolve, because then you're not talking about evolution in a single group, you have an ensemble of groups, and that makes all the difference. This is a separate slide that is about not in the previous example, I had altruism and selfishness starting at 50% and then increasing. In this set of slides, I'm talking about everybody's selfish and you drop a single altruist into the population. And a similar kind of Simpson's paradox argument helps show how altruism can increase in frequency even there. It's due to George Williams and Mary Williams. So I mentioned before that that the famous examples from Darwin in which he brings in group selection are all about altruistic traits. But here's a neat example from The Descent of Man where he's talking about group selection influencing the evolution of a trait that's not altruistic, and it has to do with technological innovation. So he speculates here that suppose there's a tribe in which some one man is more sagacious than the others, and that man invents a new snare or weapon or other means of attack or defense out of the self-interested reason of wanting the new tool to use for his own purposes. And the other members of the group see this cool new tool and they imitate it. They learn from that person. And then what happens is that all the individuals in the tribe have this cool bit of technology, and that tribe will now do better than other tribes that lack the technology. And the idea of being a technological innovator, the way Darwin is telling the story, is not an example of an altruistic trait, because being an innovator is good for you, and it also is good for the other individuals in the group who learn from you, imitate what you discover, and help the group to do better in its competition with other groups. So here's a sort of schematic picture of how and why technological innovation is good for the group and good for the individual, right-hand drawing, whereas selfishness and altruism are different. Selfishness is good for the individual, altruism is good for the group. And Darwin's point here, it doesn't require that innovators have a special gene at the eye locus that helps them be technological innovators. Genetic transmission is not essential here. It could be individuals learning from other individuals, passing technological skills and interests through the generations by teaching and learning. Okay, so this is the end of part one. Darwin invented the idea of group selection to explain the presence of altruistic traits, but he used it occasionally to explain other traits. And it seems at first glance that the definition of altruism and selfishness means that altruism cannot evolve by natural selection at all. But if you understand Simpson's paradox, I hope you can begin to see how that's a mistake. And what's key is to distinguish how altruist and selfish individuals how their fitness is related when they live in the same group versus what it's going to look like when altruistic and selfish individuals live in multiple groups. And the thing that's possible and essential for altruism to evolve is, though altruists are less fit than selfish individuals within each group, it can turn out that the average altruist, averaging across groups, is fitter than the average selfish individual. Okay, that's the first part. Here's the second part, which is on Darwin's discussion of evidence for common ancestry. This is the only figure from the origin of species. What you see here is what biologists now call phylogenetic tree. You have the present here with a bunch of objects. These could be species or individual organisms. And as you go down the page, you're going back in time, and you're seeing how objects, these three objects have a common ancestor here. These 5 objects have a common ancestor here. And the 15 objects that are at the top of the page are all descended from 1, 2, 3 individuals at the bottom. And Darwin's idea is that if you went back further in time, those 3 individuals would probably be descended from 2 or even 1. And so that's the idea that biologists now call universal common ancestry. The idea that everything alive today on Earth is descended from a single common ancestor. And Darwin's ideas, common sense ideas, the evidence for common ancestry is that is similarity. It's the fact that organisms are similar to each other that makes plausible the idea that they're descended from common ancestors rather than arising separately in evolution. So this is, I'm going to call this his first principle, that if 2 species have a given trait, this matching of the 2 species is evidence for common ancestry. And I want to think about why this is, it seems sensible, but I want to try to give a kind of mathematical framing of this idea to show why it actually makes sense. And the tool I'm going to use is called the law of likelihood, which says that observation favors one hypothesis over another precisely when the observation has a higher probability if the first hypothesis is true than it would have if the second hypothesis were true. And I'll get to the second thing a little bit later. So to illustrate the intuitive plausibility of this idea, suppose you have 2 hypotheses about the composition of this urn. The first one says that 80% of the balls in the urn are green. The second says that only 50% are green. And suppose you sampled from the urn and you observe that 90 of the balls in the sample are green. Now, this observation does not prove that H1 is true. It does not prove that H2 is true, but I hope you'll agree with me that this observation is evidence in favor of H1 as compared to H2. The observation favors H1 over H2. And if that seems intuitively correct to you, you're reasoning the way the law of likelihood tells you to reason about the interpretation of evidence. And the thing to bear in mind here is that we're not talking about the probabilities of hypotheses here. This is the probability of the observation given the hypothesis. It does not say that H1 has a higher probability of being true than H2 does given the observation. So Darwin thinks that similarities are evidence for common ancestry. If you put that into a law of likelihood framework, what you're saying is that the fact that two species have this trait favors the hypothesis of common ancestry over the hypothesis of separate ancestry, in the sense that the similarity would have a higher probability if common ancestry were true than it would have if separate ancestry were true. Okay, so now I want to talk about some assumptions that are totally standard in phylogenetic inference in biology now that allow you to show that Darwin's hunch was correct. Similarities really are evidence for common ancestry, given these assumptions. So what I've done is I've taken the common ancestry and the separate ancestry hypothesis, and I'm going to make some assumptions about these 5 letters that you see here. Just look at the ones on the left in the common ancestry hypothesis, PA, QB, and C. Each of those is a probability. It's a number between 0 and 1. So all the probabilities we're going to assume are they're bigger than 0 and less than 1. The second assumption has to do with the relationship with P and A. Let's start with that. P is the probability that X has T if this ancestor has trait T. A is the probability that this individual has trait T if this ancestor lacks trait T. And similarly, Q is the probability that Y has T given that C has T. B is the probability that Y has T given that C lacks T. And the assumption that you have under 2 is just that if this is bigger than that, then Q is bigger than B. What is that? And the reason that's a standard assumption in phylogenetic reasoning now is this. If this descendant has trait T, the probability of that would have been greater if the ancestor already had T than it would be if the ancestor did not have T. So P is bigger than A, and similarly Q is bigger than B, a standard assumption. There's an assumption called conditional independence, which I'll skip over for a moment. I guess I'll just skip it. And then there's the assumption that those PA, QB, and C guys over here are also applicable here. So the probability of X having T, it depends on C, P, and A. And the probability of X having T over under the separate ancestry hypothesis depends upon the same numbers. So here's something weird. The probability that X has T is the same on each of those 2 hypotheses, the common ancestry and the separate ancestry hypothesis. And the probability that Y has T is the same on the common ancestry and the separate ancestry hypothesis. However, here's the surprise. Given these assumptions, the probability that X and Y would both have T is greater on the common ancestry hypothesis than it is in the separate ancestry hypothesis. In other words, the probability of getting a matching is greater when there's common ancestry than there would be if there were separate ancestry. Okay, so that's the simple kind of simple explanation of why Darwin's instincts were correct about similarity. If these assumptions hold, you've got a likelihood justification for that way of interpreting similarities. Okay, now here's an interesting passage from The Origin in which Darwin is saying, well, yeah, similarities are evidence for common ancestry, but some similarities give you stronger evidence for common ancestry than others. And he says, adaptive characters, although of the utmost importance to the welfare of the being, are almost valueless to the systematist, for animals belong to 2 most distinct lines of descent may readily become adapted to similar conditions and thus assume a close external resemblance. But such resemblances will not reveal, will rather tend to conceal their blood relationships to their proper lines of descent. So what does this mean? It means that useless or deleterious similarities provide strong evidence favoring common ancestry over separate ancestry, whereas useful similarities do not provide strong evidence. They provide very weak evidence for common ancestry. So let me give you an intuitive, this is, and I'll tell you the examples that Darwin gives to illustrate these ideas. One of them is his. Dolphins and sharks are both shaped like torpedoes. He doesn't use that word. They're fusiform. They're shaped in this way. And he says that's not strong evidence that they have a common ancestor because they're both large aquatic predators, and the most fit body form for a predator of that sort is to be shaped like a torpedo. So you'd expect these guys to evolve that shape, even if they had no common ancestor. On the other hand, here's the example that I made up to sort of contrast with Darwin's. Consider the fact that monkeys and human beings have tailbones. That's more interesting because the tailbone for human beings is not useful to us. And so that's the sort of similarity which would lead Darwin to say, that's better evidence, that's stronger evidence that human beings and monkeys have a common ancestor. So how do you think about stronger or weaker favoring within the idea, within the framework of the law of likelihood? So we're back to our EARN example. H1 says 80% are green, H2 says 50% are green. Here are two different data sets you might get. And I hope you get the feel, you sort of feel the intuition that O1 is stronger evidence favoring H1 over H2. O2 is much weaker evidence. And the reason is that if you got 90 out of 100 balls being green, that would be much more probable under H1 than it would be under H2. Whereas you got that, H1 would have a tiny, tiny degree of higher likelihood than H2 has. So let's now try to write that out, using the law of likelihood to think about how useless similarities, neutral deleterious similarities, are different from adaptive similarities. So here's the likelihood ratio that represents how strongly a neutral or deleterious similarity favors common ancestry over separate ancestry. Here's the likelihood ratio that describes how an adaptive similarity favors common ancestry over separate ancestry. And each of these likelihood ratios is greater than 1. So we've already seen that similarities favor common ancestry over separate ancestry. But the idea is that the neutral and deleterious similarities favor common over separate ancestry much more than adaptive similarities do that favoring. And so from a law of likelihood point of view, you'd expect this ratio to be bigger than that ratio. There is an argument due to Anthony Edwards that Mike Steele and I built on to actually get a sort of probability feeling for why this claim down here is correct most of the time. So we have over here the common ancestry hypothesis confers a probability on that proposition. The separate ancestry confers a probability on that proposition. And then the probability that X and Y both have trait T can be written equivalently as the probability that X has trait T given that Y has trait T times the probability that Y has trait T. And the one on the bottom line, that X and Y both have trait T given the separate ancestry hypothesis, is the same as two separate probabilities multiplied together. The probability of X and Y being that way, if they don't have common ancestry, is just the one times the other. That is, they're independent of each other under the separate ancestry hypothesis. And then, given some of the assumptions I mentioned before, you can simplify this argument, and you get down to this line where the probability of the common ancestry over separate ancestry, the probability of the separate ancestry hypothesis, is going to reduce to the probability that X has the trait given that Y has the trait given P, where P is just the probability that one of those guys has the trait. It could be big or small, it doesn't matter what it is. Okay. And so this little bit of algebra that I walked through probably too quickly, it says that given some assumptions, the likelihood comparison here, common to separate ancestry, breaks down to 1 over P. And the reason where that number 1 comes from is the assumption that if X and Y have a common ancestor, the common ancestor existed very recently. So if one descendant has it, the other almost certainly does too. So that's where this number 1 comes from here. And P is just the probability that X has the trait or Y has the trait. It could be any number between 0 and 1. But now, so this is the likelihood ratio of common to separate ancestry here is 1 over a probability P. And notice that this ratio gets bigger the smaller P is. P is the probability that some descendant has trait T, X or Y has trait T, and you get this sort of neat result. Suppose trait T is extremely deleterious for the organisms we're talking about, then the probability that the descendant will have it is very low, which means that 1 over P is very big. So this is super strong evidence for common ancestry. If it's neutral evolution, the value of P is a bit bigger, and so the strength of the common ancestry, the strength of favoring is a little lower. And down at the bottom of the page, just as Darwin said, is the idea that adaptive similarities don't give you very strong evidence for common ancestry because the probability that a lineage will evolve them is very high or higher when there's an adaptive similarity, which is favored by natural selection. So this likelihood analysis basically shows you how Darwin's intuitions about how deleterious or neutral similarities give you stronger evidence for common ancestry than adaptive similarities, how it plays out and can be clarified, I hope, by thinking about likelihoods. Here's a problem for Darwin's story here. Biologists mostly will tell you that the genetic code is known to be nearly universal. And the fact that it's present in virtually all the organisms on the planet, all the species, is usually cited as strong evidence for universal common ancestry. But that's kind of puzzling given Darwin's take on how adaptive similarities are not strong evidence, because the genetic code that a sexual organism uses is extremely useful to it if it wants to reproduce, because it's got to have the same genetic code that the other organisms have in the species, or else it will be impossible for it to reproduce successfully. So this is an example of it's super advantageous for an organism to have this genetic code rather than that one, where this one is the one that everybody else has. And yet it's a useful similarity, but yet it seems to be strong evidence for common ancestry. It seems like it's totally different from the dolphin and shark example where you say, well, you'd expect the organisms to have that shape even if separate ancestry were true. So what's going on here? Okay. Okay. So how does that make sense? Well, here is a picture of a kind of selection in which adaptive similarities give you very strong evidence for common ancestry. And it's our fitness function, but it's not about altruism and selfishness now. You have selection for the majority trait. Notice at the 50-50 point, T1 and T2 have the same fitness, but when T1 is in the majority, T1 is much fitter than T2. When T2 is in the majority, it's we're down here, and T2 is fitter than T1. So what will happen in a situation is that a population will either evolve by natural selection to 100% T1, depending upon where it starts, or to 100% T2. And in this this is I hope a kind of suggestive picture. If there are two genetic codes, which is the better one to use? Well, it's the one that everybody else in your species is using. And in this kind of model, where you have these two possible outcomes of natural selection leading to stable frequencies of the trait, it turns out that the probability of these two descendants having the same, of one of them having the trait given that the other does, X having the trait given that Y has the trait, under selection for the majority trait, is the idea they would be similar is more probable under this kind of process when there's common ancestry than it would be here. And the upshot of that is that here's an exception to Darwin's principle here. When you have stabilizing selection for a trait, so if the population is at 100% T1, selection is going to keep it there. If it's at 100% T2, selection is going to keep it there. In that case, you can show that similarities due to stabilizing selection are stronger evidence for common ancestry than neutral similarities are. So this is an exception to Darwin's idea that adaptive similarities are never strong evidence for common ancestry. They are sometimes, depending like that, but he was right about this kind of case down at the bottom here. And all this is sort of intelligible when you use the law of likelihood as your technical tool for thinking about what strength of evidence means. Okay, the last topic I want to talk about here is not about inferring common ancestry, but the way that Darwin used the fact of common ancestry to test hypotheses about adaptation. It's a great example. Darwin says the sutures in the skulls of young mammals have been advanced as a beautiful adaptation for aiding parturition. That's a word in English that is never used anymore. It just means live birth. And no doubt they facilitator may be indispensable for this act, but as sutures occur in the skulls of young birds and reptiles, which have only to escape from a broken egg, we may infer that this structure has arisen and has then been taken advantage of in the parturition of the higher animals. And Darwin is saying in this surrounding passage that although skull sutures, which make your skulls flexible and squishable through the birth canal, they're useful in terms of facilitating live birth, but that's not why they evolved. And he's saying the reason for this is, I hope this puzzles you a little bit if you haven't seen the example before. He says the reason you can see that's true is because think about reptiles and birds. So this is a very puzzling passage. And you might ask the question, I want to ask the question, why are the characteristics of current birds and reptiles relevant to testing a hypothesis about why mammals have skull sutures, why they evolve skull sutures? And he doesn't really spell out the reasoning, but here's what I think he's doing. He's exploiting the fact that mammals, reptiles, and birds have a common ancestor. And he's thinking about a phylogenetic tree, something like this, where all three of these groups have skull sutures, yeses, and live birds found in mammals, but not in these other groups. And then Darwin is making an inference about the characteristics of the common ancestors shown in the tree. He thinks that skull sutures were present here and here, and that the absence of live birth were present here and here. And you see, I'm calling this a parsimonious inference. Darwin doesn't talk that way, but I am. He's, I think, inferring that these are the characteristics of the ancestors in the interior of this tree, because if that's the way the ancestors were, there would have to be fewer changes in characteristics as you move from past to present. If you had other assignments here to the interior of the tree, it would be unparsimonious because you require more changes in character state. So, for example, if you thought that there were no skull sutures down here, then you'd need to evolve from no skull sutures here to skull sutures there, no sutures here, there, and there. You need 3 changes to get the data at the top of the tree, given that assignment. But if you assume that skull sutures are already present in this lineage, you don't need any changes to occur coming to the present. And that's what makes it more parsimonious, and Darwin thinks more plausible. And because of this assignment of characteristics, what is he thinking about the lineage leading to present mammals? He's thinking that sutures were present way, way before live birth evolved. So the idea that sutures evolved to facilitate live birth is exactly backwards chronologically, because live birth was there for a long time. Sorry, sutures were there for a long time. Live birth happened later. If this is how Darwin is reasoning, I think there are other examples in which he does this. He's testing a hypothesis about adaptation by using the idea of common ancestry. And if that's one of the items in Darwin's toolkit, it helps you, I think, resolve a mystery about how Darwin changed his mind about a pretty important subject, namely the problem of how the sex ratio evolves. Sex ratio means the percentage of males and females in a species. In the first edition of this book about human evolution, Descent of Man, Darwin describes how natural selection will shape the population's sex ratio. And in the second edition, 3 years later, he says, what I said before was wrong. It's too complicated. I just withdraw my suggestion. And the frustration for Darwin scholars is to understand why he changed his mind. And he never says it. And I and other people who looked in archives, there's no evidence. It seems like he never wrote a letter to anybody about this. He just changed his mind, at least as far as anybody can tell. Why did he change his mind? Well, I think what I'll do is I'll summarize the ideas here, just for reasons of time. Darwin's theory in the 1871 first edition is that even sex ratios evolve because reproduction is monogamous. But then I think he then realized that when you look at human beings and other hominids, you see even sex ratios really across all the species in this group. But monogamy, this is human beings here. Some human societies are polygamous, polygyny, multiple females in a mating group. And some populations are more or less monogamous. But when you look at other hominids over here, they're all polygynous. And he then did what he did before in terms of inferring character states of ancestors, and got this result, and reached the conclusion that there was an even sex ratio down here with polygamy, and only as you get all the way over here does monogamy appear. So I think what he was thinking here is that the even sex ratio did not evolve because there was monogamy. Monogamy came into the picture much more recently, where an even sex ratio was around for ages. So you see, there's a kind of chronological reasoning here that would explain why Darwin thought he was all wet in thinking that the key to understanding why even sex ratios evolve is that monogamy is doing the work. So here are the topics that I've talked about in the second part of this presentation. I talked about Darwin's idea that similarity is evidence for common ancestry. I've talked about his second principle, that he thinks that adaptive similarities provide meager evidence for common ancestry, while non-adaptive similarities provide stronger evidence. And finally, there's the discussion of how he uses common ancestry to infer the characteristics of ancestors and to use that to test hypotheses about adaptation. On the first of these, standard assumptions about phylogenetic processes show you that Darwin was on the right track when he's had that first principle. The second one mostly falls into place using a likelihood analysis also, except an exception to what Darwin was saying comes when you think about stabilizing selection. And finally, the way common ancestry helps you think about adaptation is by testing for what the chronological order is in which traits evolve. If one trait is present for ages before another one appears in a lineage, you've got to make sure that your adaptation hypothesis doesn't have that exactly backwards. So thanks to my co-authors for some of this work, and thanks to you for your attention.
Well, as I was saying, I'm feeling very privileged to be invited to help celebrate Elliott's contribution to the fossil biology. I'm going to be talking more generally about some of the contributions that Elliott has made, rather than focus in on his lecture that he gave today, although I will touch on one or two issues that he raises in his recent book. But there's no other philosopher of science that I admire more, or who has influenced my thinking to a greater extent than Elliott, nor in my judgment, which admittedly is that of a biologist rather than a philosopher, is there another living philosopher of biology who's made a greater contribution to his field than Elliott. I vividly recall reading at an impressionable age Sober's classic 1984 book, The Nature of Selection. And I think it's fair to say it had a transformational impact on my thinking. Not only did I learn a great deal about evolution biology from this text, insights that could not at the time be found and to this day remain scarcely treated in biology textbooks. But the book helped me belatedly to the realization the relationship between the fields of biology and philosophy could be mutualistic rather than parasitic. In other words, that biologists could learn about biology than philosophers. I adore the clarity of thought in Elliott's writing, none of which I'm happy to say has been lost in his latest contribution. I also relish the the courage, the daring of Elliott's deep delve into some truly fundamental issues about how evolution works, which hitherto had mysteriously been given relatively short shrift by my biologist colleagues. Key points that I took from The Nature of Selection from Elliott's later books and papers, particularly those shown in this slide, include the distinctions between selection of and selection for a character, between source laws and consequence laws, and between variational and transformational explanations for change, as well as, of course, the evolutionary significance of group selection. To my mind, Sober's writings on these topics and many more have been truly foundational. They've raised the bar for work in the field of philosophy of biology to such a degree that they helped establish the field itself, not just as a subfield of philosophy, but as an adjunct to biology. So you can view my comments as an invitation for Elliott to address more directly some of the issues that developmental biology raises for evolutionary science, which are central to Evo-Devo and an extended evolutionary sequence. I'll begin with an important Sober insight. Sober famously distinguished between what he called source laws and consequence laws. To quote from The Nature of Selection, the former describe the circumstances that produce forces, the latter describe how forces, once they exist, produce changes in the systems they impinge upon. Sober characterized evolutionary theory as rich in consequence laws, the province of population genetics, but impoverished with respect to source laws. Indeed, the latter are often viewed as the subject of other fields, such as physiology or ecology. Population genetics provides valuable tools with which to track the genetic consequences of fitness differences, but this tells us little about the causes of variation in fitness. So most contemporary evolutionary science uses fitness differences to explain adaptation and phenotypic change. Not everyone is entirely satisfied with that kind of explanation, myself included. For instance, another philosopher, Dennis Walsh, points out that an evolving population simultaneously undergoes changes in its lineage structure, as some genotypes die out and others become better represented, changes in adaptiveness as the population comes to comprise individuals better suited to their environments, and changes in trait frequency, as some traits increase and others decrease in representation. Fitness is a specification of the relative rates of birth or death that particular genotypes experience, and wrapped up in that description is an implicit expectation of concomitant changes in adaptiveness and trait frequency. It's a moot point as to whether identifying fitness differences is providing a completely satisfactory causal explanation for adaptation or trait frequency changes. Some would say it's not a causal explanation at all. It's more of a statistical analysis of the consequences. Irrespective of whether or not you agree, it's certainly possible to envisage a richer causal explanation, one that explained why those particular adaptations evolved and why that particular selection ensued. So today, I invite Elliott to reflect on whether a greater focus within the field on development might help deliver such richer explanations. I'm going to focus on the hypothesis that the developmental processes of organisms create fitness differences and thereby determine the nature of selection, in the process perhaps helping to reveal what might be considered source laws for evolutionary biology. I'll consider two ways in which organisms may do this. First, organisms can engage in niche construction. That is, they can create and modify benign environments that help to keep themselves and their offspring within adaptive bounds. And second, organisms can respond to experienced conditions plastically, generating a complementarity between themselves and their environments. In simple terms, the organism can change its immediate environment to suit itself, or it can change itself to fit the environment. So I'll give you one empirical example of each to show you how these activities might determine the character of selection. So the niche construction example is provided by a lovely series on dung beetles by Armin Mosek and his group at Indiana University. They show that dung beetle niche construction directly causes and strengthens the relations between key dung beetle traits and fitness. The mother beetle builds a brood ball out of dung. She lays an egg in it on a pedestal containing a microbiome, and then she buries the ball underground. The larvae then eat the pedestal, thereby acquiring valuable symbionts, and process the brood ball in various ways, including constructing a pupation chamber, as well as feeding, defecating, and mixing the materials, which redistributes the inherited microbes, and thereby allows the developing larvae to extract carbon substrates more efficiently. So all of these activities are examples of beetle niche construction. Mosek's team conducted experimental manipulations that either permitted or cancelled the environmental effects of this niche construction, for instance, by removing the pedestal or regularly relocating the larvae into fresh dung, and in this way showed that the modification of environments by mother and larvae were critical to normal beetle development. Niche construction not only substantially enhanced larval growth and adult body size, but was also what determined the relationship between key secondary sexual traits, including foreleg size, eye size, and horn size, and fitness. Beetle niche construction not only shaped phenotypic variation, but in the process dramatically altered the relationship between some key traits under selection and fitness. For instance, if we want to understand why horn size is under sexual selection as opposed to any other beetle character, the answer is that this trait is unusually sensitive to the manner in which larval niche construction makes carbon nutrients available to the growing beetle and is thereby an honest signal of beetle quality. Such studies provide explanations for why those particular traits have been favored by selection. What selection occurs is largely determined by beetle niche construction, and it's in this sense that we can regard niche construction as a cause of selection. The example of adaptive plasticity concerns blind Mexican cavefish, Astyanax mexicanus. When these fish were first discovered in 1936, they appeared so different from other known fishes that they were listed as a new genus, only for subsequent research to reveal that their cited surface-dwelling conspecifics are actually widespread in the rivers and streams of Mexico and southern USA. At least 30 cavefish populations are now known, mostly eyeless and with reduced pigmentation, but also sometimes exhibiting enhancements in non-visual sensory systems, as well as changes in bone structure, fat storage, starvation resistance, metabolic rate, and foraging behavior. Intriguingly, when Helena Belanzier and colleagues at the University of Maryland raised surface Astyanax in complete darkness, they found that these fish exhibit many of the traits of their blind cavefish conspecifics, including changes in the thickness of the retina, increased resistance to starvation, decreased metabolic rate, and enhanced fat storage. Hence, precursors to many cave-related traits appear in surface fish within a single generation through phenotypic plasticity. Moreover, the same patterns of correlated variation in developmentally codependent morphological, physiological, and behavioral traits in response to dark rearing was repeated in multiple experimental populations. The fact that different populations respond similarly explains why a virtually identical response to natural selection was manifest in over 20 independent cavefish populations. There's an impressive regularity to the plastic response, a foreseeable pattern to the trait variation, fitness differences, and trait heritability arising in the population, which leaves the eventual evolution of the blind cavefish phenotype highly predictable. As we saw for niche constructing beetles, here the developmental plasticity of these fishes co-determines the nature of the response to selection by generating, in a surprisingly well-regulated manner, a developmentally biased distribution of correlated phenotypic variation. In both the beetle and the cavefish examples, I'm inviting Elliott to consider the possibility that developmental regularities in patterns of correlated variation subject to natural selection might be considered source laws for evolutionary biology. So I want to pin these examples down. I want to use them to explore how developmental processes might be shaping evolutionary dynamics. And I'm going to try and illustrate that role graphically. So these axes represent dimensions of phenotype space. They could, for instance, represent the height, width, and depth of a morphological character or its developmental timing. Of course, in practice, phenotype space is highly multidimensional, but for our purposes, we'll restrict ourselves to 3 axes. And this blue line represents the range of variation of a trait in that phenotype space, which for simplicity, I will represent as linear, although in reality, it will be a volume of phenotype space. This can, for instance, represent variation in beetle head size. We can also imagine that there's a frequency distribution associated with this trait, such that there are more individuals with intermediate values and extreme values, and that there's a fitness function associated with it, such that some of these trait values have higher fitness than others. In reality, of course, such distributions will vary with the developmental environment. But for my purposes today, all you really need to remember is that there's variation in the phenotype and in fitness along this dimension. And we can also imagine that there are other traits in the organism that co-vary with this first trait. The size and shape of left and right legs will be correlated, as will forelimb and rear limb size. Quantitative genetics theory has established that traits that are selected together because they are functionally related can, through the selection of pleiotropic variants or epistatic interactions, become genetically and developmentally integrated. So gradually, functionally and structurally related traits will evolve into a cluster of correlated traits, such as shown here. Because these traits are correlated and because all contribute to fitness, we can think of this cluster as a single major dimension of correlated trait variation. Evolutionary geneticists have studied the dimensionality of genetic variation in dairy and beef cattle, in mice, in fish, and in fly populations, often finding that selectable genetic variation lies in a small number of such dimensions. And recent thinking is that much evolvability is bound up in a few underlying dimensions of correlated variation that collectively have strong impacts on fitness. It's largely because of this developmental covariation that it's easy for biologists and breeders to select for any isolated trait, but not for any combinations of traits. We're now in a position to specify what niche construction of phenotypic plasticity are doing here. In the case of the blind Mexican cavefish, for instance, the plastic response to the cave environment shifts the cluster of variation that dominates fitness in the surface river environment, shown here, to the cluster of traits that dominate in the cave. No, I'm not just saying that selection pressures and therefore selected traits change in a novel environment. I'm saying that how they change is determined by the plastic response. The cluster of traits that dominate fitness in the cave environment are the very same cluster of traits that experiments show exhibit increased variation when surface fish are reared in the dark, as well as other traits correlated with them. Both that plastic response and the evolutionary response of cave populations has been found to be highly repeatable and strongly correlated. Plasticity is doing two things here. First, it's generating the new cluster of correlated traits. What these traits in red have in common, what makes them correlated, is that they're all developmentally linked. They all respond plastically to the dark environment. They're connected by shared developmental regulatory mechanisms, often comprising shared gene regulatory networks. They're all pretty well researched. But secondly, plasticity is shifting the dimension of trait variation that dominates fitness. And beetle niche construction is doing the same two things. It too is generating a new cluster of correlated traits, tying together the fate of behavioral traits in the mother, such as brood ball manufacture and burial, with behavioral traits in the larvae, such as brood ball processing, with morphological traits in the adult, such as horn size, with morphological traits in the larvae, such as growth. And it too is switching the dimensionality of trait variation that dominates fitness to the resulting novel clusters of traits. These studies also show how selection on the reaction norms created by niche construction allows populations to evolve rapidly along developmentally biased pathways, representing these clusters of mechanistically and functionally associated phenotypes. Evolutionary biologists, of course, have got used to thinking of reaction norms as genetically determined, but this is a convenient heuristic, not a biological fact. In reality, reaction norms are the product of developmental systems. And I've thrown in a third example, domestication syndrome in mammals, just to make the point that there's nothing special about plasticity and niche construction in this regard. Without either, there can still be developmentally biased phenotypic variation organized into major clusters of phenotypic traits that dominate fitness. It's because of the existence of that cluster of correlated phenotypic variation that selection for tameness in a variety of mammals also led to the evolution of a bunch of other traits, such as curly tails and piebald coloration. What links the very odd assortment of traits characterizing domesticated species is shared developmental regulation by neural crest cell GRNs. So in the nature of selection, Sober also made the distinction between selection of and selection for a character. The same point is made in Elliott's new book manuscript. In both books, to illustrate his point, Elliott introduces this children's toy, which is a container structured into levels by dividing partitions and containing balls of different sizes and colors. If the balls are initially at the top, shaking the toy eventually redistributes them throughout the toy, with the smallest balls traveling furthest through the holes to the lowest levels. The smallest balls are the objects that are selected, but red balls have been selected at the same time. Sober stresses how there is selection for smallness and selection of redness. Well, helpful though this analogy is, there are important respects in which the selection toy, I think, gives a misleading impression of the action of natural selection. A characterization that portrays organisms in overly passive terms. For instance, the structural features of the toy, the balls and the partitions, were factory made. The balls played no part in building the partitions. However, we've seen that beetles construct and process brood balls, switching selection away from one set of traits and towards another set of traits. It's the action of living organisms that determine what is selected for. It's as if the balls in the selection toy build the partitions and cut out the holes themselves. Furthermore, the balls in the selection toy are solid, inflexible objects that move only when acted upon by external forces. Yet, as we've seen, in the real world, animals such as the Mexican cavefish can respond through adaptive plasticity, adjusting their phenotype to match the world. It's as if the balls are made of a squidgy jelly and are able to squeeze through tiny holes. Finally, in the selection toy, the association between ball size and color is entirely arbitrary. There's no reason why the small balls needed to be red. We know the toy could just as easily have had small green balls. And this subliminal knowledge gives the impression that in selecting for smallness, redness has been randomly chosen. But here again, the analogy is misleading. Floppy ears are not linked to the tameness of domesticated animals by chance. These traits and other components of domestication syndrome are connected by joint developmental regulation through the neural crest GRN. In the real world, developmental mechanisms connect the selection of traits to the selection for traits. Note, in making these points, I'm not criticizing Sober's analysis. Sober's selection toy really does provide an excellent analogy for how evolutionary biologists study natural selection. The sources are taken for granted and the consequences are the focus. Rather, my point is that evolutionary biologists' focus on masks hidden determinants of the sources of fitness differences. In the real world, small balls are red for a reason. So these issues are relevant to topics that are discussed by Elliott in his latest book. For instance, in the chapter on mutation, Elliott presents and critiques Fisher's geometric argument, illustrating why Fisher believed large mutations were less likely to be adaptive than small mutations. I actually find compelling Elliott's claim that Fisher's argument falls down when one also considers the probability of fixation, which is greater for large mutations. However, there are other relevant assumptions at play here that, in my eyes at least, further undermine Fisher's claim. Fisher is assuming that the effects of the mutation are equally probable in all phenotypic dimensions, and hence that the mutation shifts the phenotype to a point in a circle, since phenotypic variation is unbiased. Conversely, in the real world, as we've seen, phenotypic variation is often biased into these developmentally linked clusters, with some combinations of traits co-occurring, such as when cavefish acquire enhanced starvation resistance and fat storage along with eye loss. That's equivalent to turning these circles into ovals. These figures from a recent paper of mine present a plan view of an adaptive landscape where the contours represent fitness, the plus represents the optimal phenotype, and the shaded regions represent the distribution of novel phenotypic variation. Well, following Fisher, the default assumption in evolutionary theory has been that the distribution of evolutionarily relevant phenotypic variation introduced into the population is unbiased, as in the top panels. Hence, we go back to circles. However, there is now good empirical and theoretical evidence that ancestral selection biases variability in dimensions loosely aligned with the direction of past selection, as in the bottom panel, undermining Fisher's geometric argument. Novel mutations are not so much guided towards the optimum as directed along dimensions aligned with ancestral selection's steep fitness gradients. Often, but not always, that orientates the long axis of the oval loosely towards the optimum. Such developmental bias may increase evolvability and make it more likely that adaptive phenotypes arise. For instance, mutations might increase or decrease the length of both left and right legs, perhaps with correlated effects on both arms, but are less likely to generate left and right legs of different length. That's because past selection of longer or shorter limbs strengthen the epistatic interactions between genes that produce useful symmetrical correspondences in limb length, while reducing the strength of gene interactions that produce dysfunctional asymmetries. It's in this sense that useful or facilitated variation is produced. Natural selection shapes developmental machinery in ways that alter the pleiotropic effects, such that each mutation generates well-coordinated changes in an integrated package of traits. In a stellar career, Elliott Sober has not only conducted some of the most insightful analyses of evolutionary phenomena, but also provided key conceptual tools with which to dissect contemporary debates within the field, including how development affects evolution. It would be wonderful to see arguably the world's best living philosopher of biology sink his teeth into these weighty issues. It remains me to thank my collaborators on this work, Tobias Uller, Natalie Feiner, Mark Feldman, Scott Gilbert, and you for your attention. Thank you.
Okay, first of all, thanks a lot for inviting me to this special occasion, and again, all my congratulations to Elliott for his award. I have to say that personally, my meeting group selection has been really the tipping point to let me decide to switch from physics, where I was educated, into evolutionary biology. So the perspective I would provide today is, as you will see, not that philosophical and maybe also not all that biological, but I hope it will be useful anyway. It works. So how do I take this away? Sorry. Yeah, just because otherwise you can't see my titles. Okay, great, thanks. So in my main interest in my research is how eco-evolutionary feedbacks take place between organisms or particles that are represented. I think the laser pointer is not working, but okay, well, that are represented here on the left. And now this is okay, yeah, here. So these particles or organisms, they differ in some traits, and the frequencies of these traits change in time in a whole population. Particularly, I am interested in knowing how is this process affected by the existence of an intermediate level of organization that I will call a collective level that could be, for instance, groups. And particularly, I deal with populations that are made of cells, and this collective level of organization is represented by multicellular organisms, including those that only exist for one generation. I'll tell you more later, and microbial communities. So my talk is organized in two parts. In the first part, I would like to come back to the Simpsons paradox and introduce some formalism to understand what is the effect of structure into groups on statistics at the population level. And later, I would like to address how the architecture of group genealogies affects selection. So let's start with a very schematic representation of how a population of units or of individuals or particles, as you want to call it, may look like. So on the left-hand side, you have these individuals, and you can imagine that they are partitioned into groups. For simplicity now, I'll take groups of the same size, so that each group of index k is characterized by the frequency of particles pk. So let's take as a reference blue particles, and let's say that we start with a population that has a fraction p of blue particles. Sorry, I don't know what happened. Yeah, exactly. It went straight to the end. Okay. So what we want to know, or we know sometimes in advance, is what is the initial proportion of the blue particles. And this proportion, in the case where groups are all of the same size, is simply the average proportion within each of the groups. Now imagine that if you start with a subdivision, so certain groups of the same size at time t, you have some process by which groups change in time, and therefore you observe again the same groups at a different time t prime. So the groups in going from tau to tau prime, they can have changed in the size and in composition. And if you want to compute what is the average fraction of blue types at time t prime, you have to take into account the fact that the proportion within each group is multiplied in this sum by the number of individuals of the group. So what one is really interested in is to know how does this proportion change from tau to tau prime. So imagine that the blue type is a cooperator type, then that would mean that within groups, the proportion of blue individuals is expected to decay over time. Does this necessarily imply the fact that the average also decays over time? Well, if groups don't change in size, then this factor will be 1, and then the proportion at time tau prime will never be larger than the proportion at time tau. But if you allow between group dynamics so that groups that have a larger proportion of blue individuals grow faster, then this sum can, in certain circumstances, be larger than 0. And this is what is called the Simpsons paradox. So what does the Simpson paradox say? So it tries to, it answers to this question, is it possible that P prime is larger than P, knowing that within each group, P prime K is smaller than Pk? And the answer, as I was mentioning, is yes. Not only it is possible, but this has been also demonstrated experimentally in this very elegant work, where people have shown that bacteria that produce colicins, so a public good, can evolve over repeated repeated dilutions. The question then becomes when and how does this happen? I want to stress that this equation that is just a mathematical equality says nothing about three important things. First of all, what are the appropriate times, tau and tau prime, at which one should observe the metapopulation? It says nothing about what is the relation between a group that has index k at time tau and at time tau prime. And it also says nothing about where do groups come from. They can come from self-organization, from niche construction, ecological scaffolding. There can be many different ways groups are generated. So this brings me to the fact that one can have alternative views on what is group selection. For what I understand, Elliott's point of view is that group selection occurs every time the population structure generates variability in group productivity, so that the Simpson's paradox applies. But one can also say, okay, this is maybe too general, and one could restrict the group selection to a case where groups are effectively Darwinian individuals. And now to explain what is a Darwinian individual, let me go back to the conditions that Lemoine proposed as necessary for the evolution by natural selection. So he said that in order to have evolution by natural selection, you need to have a heritable variance in fitness. And if you think about fitness in the case of groups, then the concept of fitness and how to measure fitness might be problematic and will depend on the way you look at groups and particles. My starting point is that fitness is a proxy for demographic differences, so birth and death rates, as was also argued by Michael Döbeli and colleagues. And if you think in these terms, then the problem here is to determine how are groups born, how do they die, and especially on what time scale these processes happen. And these are not easy questions to answer if you think of some multicellular aggregates that seem to have a function, like, I don't know, biofilms, for instance, but that don't have a particularly well-established parent-offspring relationship. For this reason, with Paul Rainey, we have proposed to generalize Lemoine's conditions to cases that do not require strictly the presence of a parent-offspring relationship. And in this more general terms, one can say in the spirit of group selection that evolution can occur by virtue of group-level selection whenever there are populations of repeatedly identifiable units. It can happen. It doesn't need to happen. This leads to a following question that what does it mean that there are repeatedly identifiable groups? So there are many different ways by which groups can appear over and again in time. And I want here to point out to two extremes in the range of possibilities. The first extreme is that you start from groups that are selected, and then you take all the particles that belong to the groups, you mix them in a common pool, and then you reform groups later. This corresponds more or less to what happens in aggregatively multicellular organisms, such as the Clostridium discoideum, that is the amoeba we work with in the lab. And it also corresponds to concepts like coming together or migration pool protocols for the evolution of microbial communities. In these cases, group selection can make group properties evolve, even though there is no genealogy among groups. However, probably the possibilities that are achieved by evolution or by natural selection in this case are limited with respect to what happens when you have a properly well-established genealogy at the level of groups. So this is what can be imposed externally to communities, for instance, by serial transfer or what is called the propagal pool protocols in microbial community evolution. But it is also pretty realistic in the case of macrobiota, who are somehow linked to the, if they are vertically transmitted, to the genealogical structure of their hosts. In these cases, evolution selection at the level of the collectives is much more potent, and it can drive many, many interesting adaptations, including, for instance, the evolution of collective level inheritance that is associated to the emergence of development that is of, let's say, ecological coordination among units within a group. And in the case where you have many, many species that are interacting and that whose interactions can evolve, it can drive some specific signatures in the matrix of interactions among the species that emerge on very long evolutionary times. What is really important in these processes is the existence of different timescales. One ecological timescales that describes what happens within groups, and an evolutionary timescales that describes what happens between generations of groups. And so these conclusions on timescales, they are mostly addressed to the students of the philosophy school that is going to take place this week, because that's what we are going to discuss about. And I would like to stress that absolute fitnesses, if seen as demographic rates, are necessarily associated to a given timescale. So these timescales, they can be externally imposed, for instance, in artificial selection, or they can actually arise from the life cycle of the collective stage. However, they are really important to understand that fitnesses at different scales can be reconciled if they are looked at on the same timescale. Finally, ecological or developmental dynamics is tightly linked then to evolutionary dynamics, and the fact that they must coincide on the timescale of one single life cycle also means that the proximate and ultimate causes need to be reconcilable at this level. So the big question for me is how does one find what is the most relevant timescale to describe the evolution of a given collective property in cases where life cycles are not already very clearly established. So I would like also to conclude on group selection, saying that so evolutionary consequences of within group conflicts on long time scales, as we saw, depend on population structure. This structure can be exactly as the time scale can be exposed from the outside, or it can be generated endogenously in the evolving system. What is really important and interesting is that this structure is not necessarily fixed once and for all, but it can evolve itself. And this is the subject of evolutionary transitions in individuality that aim at understanding how does one go from, for instance, from free living organisms to organisms that are organized in multicellular groups. For the way I understood it, group selection, how Elliott proposes it, establishes this fundamental role of structure. But now, if we want to go further, I think what is really an important thing to understand is what kind of structure is realized in a given system and how it is realized. So to have a mechanistic understanding of what happens in different situations. And my personal hope is that even if one needs to do this, to look at many specific cases, there might be some general organizing principles that allow to understand at least how evolution of functionality at higher levels has evolved in certain classes of organisms. Say, for instance, when you have a single cell bottleneck versus when you have aggregation dispersal life cycles. And with this, I would like to thank my co-workers, specifically Viviane Doucier, whose work has been cited several times in this presentation. And thank you very much for your attention.
I probably go the furthest back with Elliott. I think I first met him perhaps 40 years ago in Los Angeles. I think we went to a Starbucks, which was new that time, and Elliott helped me buy an espresso machine, which I had never had before. So that was a great thing, and I've sort of been friends remotely with him ever since. So I'm pleased to congratulate him on the award, which I think is very well deserved. It's certainly the case that I read The Nature of Selection many years ago. I've forgotten much of it, but it was very impressive to me at that time. So I did read parts of Elliott's book, but not all of it, because it's a little bit mathy for me. And I've singled in on the footnote, footnote 25 of chapter 3, because that addresses, in fact, a paper that we wrote and which I had sent to Elliott a couple months ago. And that paper was by Chris Lane, who's a philosopher of ecology, and Joe Belaski, who's a mathematical population geneticist, and me. And it was a critique of a paper by Tom Whittam in Annual Reviews of Ecology and Evolutionary Systematics in 2020, which was sort of a review of the field of community and ecosystem genetics, which we thought was wrongheaded. And so I sent that copy to the copy of that paper to Elliott, and he footnoted it in chapter 25 and in chapter 3, footnote 25. And so that's what I'm going to address. So it's a very small bone that I have to pick with him. And what I'm going to say is that, yes, he is right that we were wrong in a sense, but I think we can fix that problem, the problem of wrongness, and that we can then carry on with Elliott's analogy a bit further. And I think it would be interesting to do that. And it has interesting resonances with what Silvia just said, actually, too. So this is the critique, the footnote, sorry, the footnote that Elliott mentioned in chapter 3. And that's the whole footnote. Okay, I'll just read it. Lane et al. Argue that the evolution by community selection, in the sense described here, rarely, if ever, occurs in nature, owing to the fact that communities lack heredity. The problem they have in mind arises when migrants from one generation of communities form a global migrant pool from which the next generation of communities is established by random sampling, somewhat like Silvia just said. They say that communities lack heredity in this case. In fact, a daughter community of size n is descended from up to n parent communities. This resembles biparental inheritance in the case of sexual organisms. Lane et al.'s argument reprises Maynard Smith's argument that group selection can't produce evolution because groups don't have heredity. Sober and Wilson replied to Maynard Smith by focusing on the concept of heritability. I'm not going to address all of this footnote. So I'm really going to address the parts that I'm indicating in red here. And so Elliott says, they say that communities lack heredity in this case, and I think we probably did say that. But what we actually meant was that communities lack an inheritance mechanism, so that there is no mechanistic sense by which communities give rise to offspring communities, and I think Whittam et al. Admit that there isn't such a thing, and try to gloss over it as if that wasn't a problem, which I think in our critique of Lane et al. We said that it was, and this sort of we were sort of wrong in the sense that Elliott does say it is right. Communities, but communities do have an inheritance mechanism, albeit a very complex one. So I'm going to try to explain that now. I'm somewhat confused here. In fact, the daughter community of size n is descended from up to n parent communities. This resembles biparental inheritance in the case of sexual organisms. So what Elliott meant by that is shown on the next slide. And Elliott was right, but there is a resemblance, but there can be too many parents. So here's what I'm going to say. That Elliott was right in the case of sexual organisms, that there are not too many parents. There's in fact only 2 parents for a sexual organism. But that Whittam et al. Are wrong in the sense that there are too many parents when it comes to ecosystems. So what's described, the Whittam et al. Particularly focus on the ecosystem that is represented by cottonwoods and the various insects that live on a cottonwood, and there are many species of insects that live on the cottonwoods, plus many more species of microbes that are associated with the ecosystem, which is the cottonwood ecosystem. And we're arguing that there are too many lineages, too many parents, basically, in that case, so that though there's a slippery slope between 2 parents, which is not too many, and too many parents, so that there's no right or wrong about what too many parents is, that there can be nevertheless too many parents at some point. And what do we mean by too many parents? And so this is what my second favorite philosopher of biology, Peter Godfrey Smith, wrote in 2012, and I've quoted this from many times. There must be some parent-offspring similarity, and the clarity of a parent-offspring relation of the relevant kind is inversely related to the number of parents. If there are too many parents, there are no parents at all. So that seems obvious in some ways, but why is it true? And I think it's probably true because of this. It's always phenotype that drives evolution, and if genes or alleles ultimately determine phenotype, as long as they are randomly recruited and the phenotype of the offspring is the average of that of the parents, there can be too many of those parents. And at any one locus, I will talk about that more because I think that's an important feature, which is why it's red, underlined. And the law of large numbers will intervene. So what do I mean by that? And what is the law of large numbers? And the law of large numbers, a simple statistical statement. In probability theory, the law of large numbers is a theorem that describes the results of performing the same experiment a large number of times. According to the law, the average of the results obtained from a large number of the trials should be close to the expected value and tends to become closer to the expected value as more trials are performed. I take this as an argument also about sample size, so that the larger the random samples, the more similar those random samples become to each other, and the more like the average of all the parents. So that basically the model, which I will show in a minute, that Elliott and we also have in mind, is vulnerable to that too many parents accusation. So the problem I have in mind arises when migrants from one generation of communities form a global migrant pool from which the next generation of communities is established by random sampling. And yes, indeed, that is indeed what we have in mind. And that's shown in the next slide, so that we would imagine, for instance, that parent collectives, so let's talk about communities as if they were collectives and they comprise particles, which are either organisms or the species, the species to which those organisms belong. And they periodically release all their particles into a global migrant pool, what Elliott refers to. And then the next generation of collectives is recruited randomly from that pool. It's nevertheless the case that you can retrace the reproduction of those collectives to the first generation of collectives to the second generation of collectives, so that you can do this obviously by keeping track of the particles. So for instance, particle 14, which is represented in two different collectives, gives rise to a particle, an organism of species, for instance, that is of, it's called 14, and it's labeled by 14. And then that winds up in two subsequent collectives by random recruitment. So basically the random recruitment of the offspring from the parents will produce some kind of parent offspring lineages, and therefore you can define lineages. And Elliott is right that it's not really different from the situation in which there are only two parents, for instance, a sexual organism where there are two parents, and a community or an ecosystem where there are many parents. But at some point there become too many, and why is that? And why is that has to do with Lewontin's recipe, which Silvia referred to previously. And in Lewontin's recipe, Lewontin's description of evolution by natural selection. Let me read the bottom part first. These three principles embody the principle, which I think are separate. Principles embody the principle of evolution by natural selection. While they hold a population will undergo evolutionary change, it is important to note a certain generality in the principles. No particular mechanism of inheritance is specified, but only a relationship between the parent and the offspring in terms of fitness. The population would evolve whether the correlation between parent and offspring arose from Mendelian, cytoplasmic, or cultural inheritance, which I think is what Elliott too would say is understood to be Darwin's principle. But I think Lewontin meant for the heritability, the variation, and the fitness to be separate components, separate ingredients that contributed together to his recipe, and that all three of them must be present for evolution by natural selection to occur. So the question is whether it's the heritability or the inheritance that the principle of heredity, which is jeopardized by the too many parent situation, or the principle of variability. And I would argue that it's the variability that is just jeopardized. So the variation in morphological, physiological, or behavioral traits among members of a species, the principle of variation. And what I've argued is that when there are too many parents, it's basically the case that random assembly of particles that represent the next generation of collectives gets more and more similar to each other because of the large numbers. And you know, it's a simple example would be, you know, if you wanted to look at the frequency of whether or not smoking causes cancer, you could either examine one individual, in which case that would not be enough. I mean, he might or might not get cancer as a result of it, but you could also imagine 1,000 individuals, in which case you'd get very close to the frequency with which cancer, smoking actually causes cancer. So I think that that's variability of the sample size. So it's actually variation that suffers. And because variation suffers, then that means that heritability also suffers, because there's actually no variation to be inherited if heritability suffers. So I think Elliott was right in the sense that we emphasized in our critique of Whittam et al. That it was heritability that suffered, and in fact, it's actually variability that suffers, and then heritability suffers as a consequence of that variability suffering. At least that's what I'm thinking now. So to get back to, or circle back to the principle of too many parents. What does that mean? What does that actually mean? And I've done some serious thinking about this recently, and I realized that it really means trait by trait for organisms. So there must be some parent-offspring similarity, and the clarity of a parent-offspring relation of the relevant kind is inversely related to the number of parents. If there are too many parents, there are no parents at all. But I think what Peter Godfrey Smith actually meant by that was that there can't be too many parents for any given trait. So what I mean by that in case of organisms would be, let's say, referring to Mendel's peas again, there's really no averaging of wrinkly and green. There's no conceptual way that you can average those two different traits of an organism. So I think what Peter Godfrey Smith meant was that there can't be too many parents for any given trait. And in fact, there are not too many parents. There are only two in the case of organisms, so it's not too many. So even if there's scrambling by recombination over several generations for sexual organisms, there are only two parents for any one genetic locus at any one time. So there is not too many parents on a trait by trait basis. So to get back then to the problem of community evolution, which is what the critique by Lane and I and Joe Walowski addressed, how do we do that? How can we imagine community selection to be comparable to organismal selection? And I'll just read this. For ecosystems or communities, collectives, there is similarly no averaging of herbivore and carnivore or of nitrogen fixation and photosynthesis. So a community has a variety of different traits, each of which can have too many parents or not too many parents. So how do we then reduce the number of parents? So so I would say, so what works for ecosystems is that although there may be many taxa that can perform a particular function, related or unrelated taxa, but functionally the same thanks to gene transfer. So two closely related taxa will of course likely have the same functions because they're likely to have the same genes, but it's also the case that lateral gene transfer can transfer the same function, as it were, between remotely related taxa. But only one can, because of competitive exclusion, occupy a particular or perform a particular trait for a particular ecosystem. I think that's questionable inference, but it's part of what I'm basing it on. So this function then exists really basically in two allelic forms. It can either be performed or it cannot be performed. And those are the equivalent of two alleles at any locus. So in other words, there are two parents at any locus, just as there is for organisms. That communities are not different from organisms in that sense. Because that function can be performed or not performed by the community. Different species and ecosystems are like different trait determining loci arrayed along a chromosome, in other words, and each species can either be present or absent. So I think this is sort of a new idea. I think that Joe Walowski, one of my co-authors on that paper, also had this idea, but in a different, coming from a different place. And I think that Mark Velland, an ecologist, also had somewhat like this idea when he said in a 2010 review article, in the same way that selection may favor allele A over a big A over allele little a within the species population, selection may favor species X over species Y in a community. So I think that's sort of comparable statement. And I have to do more reading about what Mark Velland has subsequently said about that. So I think that basically what we're saying, which I think Silvia did not say, was that we can in fact regard ecosystems as if they reproduced. They just have too many parents. And the problem with too many parents is not, in fact, the reproduction, because we've solved that problem. There can be as few as 2 parents for any given locus in a community, any given trait that the community might have, therefore. So that the too many parent problem can be solved in both the cases of communities by that trick, and also in the case of organisms by the trick that by the obvious trick that there are only 2 parents in a sexual species. Therefore we can come up with a reproductive analysis of the fitness of communities similar to that of the fitness of organisms. So I don't think Whitman at all, and nor do I think Elliott goes this far. And or actually Joe and I have much work to do on that system. So I want to thank Chris Lane and Joe Balowski for their contributions to this, and that's all I have to say. Thank you.
Okay, now so I'll begin with Kevin's comments. Kevin says, quotes me as characterizing evolutionary theory as rich in consequence laws, which is the province of population genetics, but impoverished with respect to source laws. I wouldn't say impoverished anymore. There are many models that describe the sources of fitness differences, and there is lots of observational evidence that document the sources of fitness differences also. But there's a special question I'm going to get to in a bit about whether there are source laws. Kevin also cites with approval Dennis Walsh's way of breaking down what evolution by natural selection involves. And notice that Kevin is treating adaptedness and fitness separately. I think that the distinction that Dennis is drawing is definitely one that I want to draw too, but my terminology, like that of a lot of people in philosophy and biology, is a bit different. What I think is needed is a distinction between an organism's actual degree of reproductive success versus an organism's ability, often called a propensity it has, to survive and reproduce. This is like a coin's bias is characterized by a certain probability it has of landing heads, and that's distinct from the actual number of heads and tails it produces in some number of tosses. Those are conceptually different, and it's important to keep them separate. And it's not just organisms that need to have this distinction drawn. Traits, which to me are the fundamental objects of biological theorizing, they have fitnesses. And one needs to have a propensity-like concept of a trait, and also a datum on its actual performance in terms of increase and decrease in frequency. So the terminology I think that Dennis is using here is, to me, not standard, but I like what he calls adaptiveness is how I use the term fitness. That's cool. That's fine. Here's a real disagreement, and I don't think it's at all terminological or not, which is, and maybe Dennis has changed his mind about this, but a while back, he was defending this view called a statisticalist view of natural selection, in which natural selection does not cause evolution. It's a way of bookkeeping and describing evolution, but it doesn't explain it. And I really think that that's a mistake. That natural selection is, to me, a cause of evolution, just like drift and mutation asymmetries and so on. Source laws versus consequence laws. Example from physics, just to ground what this distinction is about. F equals ma is Newton's consequence law. It says that acceleration is a consequence of the existence of a force. And in contrast, the law of gravitation is a source law. It tells you where a kind of force comes from. There's the force due to gravitation that pairs of objects have, which is a function of their masses and the distances between them. And now I get to the thing I want to talk about concerning laws. What I think a law is, and this is a real standard and kind of old-fashioned idea in philosophy of science, a law is an if-then generalization that describes not just what actually happens, but what can and cannot happen. It has a kind of necessity property that's, I think, very important. And typically, laws don't entail the existence of physical objects. So, I mean, the law of gravitation does not say that there's any mass in the universe. It just says what would happen if there were mass in the universe. And that's part of its generality, I think, that it's open on sort of what's actually out there, if anything. So a statement of the form that a given population has a given trait at T1 and a different trait at T2, to me, is not a law. It has the wrong logical form. What's closer to being a candidate for being a law is a statement of the form, for any population, if it has trait X at time T1, then it will have trait Y at time T2. Maybe that's a law. There can be probabilistic laws, like I have here. I mean, I think of some of Kimura's models for neutral evolution have the form, the probability that a population, for any population, the probability that it will have property T, property Y at T2, given that it has property X at T1 has a certain value. So those are laws. These are, of course, the one I'm describing here now is a consequence law. Well, let me take that statement back. But here's the thing that I want to say about laws generally, whether they're source or consequence laws. I think that it's a surprising fact about evolutionary biology and population biology, maybe more generally, that there are consequence laws that are a priori. They're not empirical statements at all. So let me give you an example of what I'm talking about here. Here's a simple example. You want to explain why some allele in a population is changing frequency, and you cook up this story. So you say in this population, heterozygotes are fitter than homozygotes at some locus. And then you trot out the usual introductory course, one locus, two allele model of heterozygote superiority, which says that if the heterozygote is fitter than the others and no other forces affect what's going to happen, then the population will evolve to a certain frequency that you can predict from the fitnesses of the three genotypes. And so this is a sort of standard little story you can make up in elementary population genetics. But the thing that I find philosophically interesting here is that the model that is on this red line here, I think it's a priori true, that if you have a population with one locus and two alleles and selection is working this way and nothing else is going on, then this is what will happen, or will happen with high probability. And to me, this makes evolutionary biology look a lot different from other sciences where the laws are actually empirical statements. I think there also are source laws that are a priori in evolutionary biology. So Hamilton's model of sex ratio evolution has this same form. It's a mathematical truth. Moore's stuff on neutral evolution is too. I'll skip this slide. Here's something from Kevin's talk that I'm surprised to hear that this idea has legs, but I'm open to learning about it. He says at one point, novel mutations are not guided towards the optimum, but rather directed along dimensions aligned with ancestral selection and fitness gradients. So let me describe how I'm understanding this. Maybe this isn't quite what Kevin was getting at, but anyhow, here's what I'm thinking as an example. So suppose a lineage of bears experienced persistent selection for longer fur because the climate these bears were in kept getting colder in the past. And this causes the probability of mutations that produce longer fur to go up in the present. So it's a kind of guided mutation that's based on the past selective history of the lineage. So how would you test this? Well, you would need time series data to say, let's look at the mutation rate of various put it along back. And if it did, you would say, oh, this is the kind of pattern that Kevin's talking about here. But there's an additional thing that's needed here. The fact that you have lower mutation rates for thicker fur earlier and higher mutation rates producing thicker fur later, that doesn't establish that there's a causal connection here. That would be the amazing part. I mean, I bet there are populations in nature that have this pattern, that the mutation rate is low at one time and it gets higher at another time, but it's the because thing that is the wow idea here. I mean, if that could be documented, that would really be, as you said, contrary to the idea of random mutation. It's guided mutation in the sort that Asa Gray wanted, though, of course, Kevin's not bringing in God here, but other than that, it's guided mutation. Okay, next, Sylvia's comments. The title of the talk is, I think, points to something very important, the evolution of collectives and collective level selection. That's an important distinction. And the idea that this is pointing to for me is that a trait that benefits the group can increase in frequency without it being true that the increase occurred because it benefited the group. And the terminology that's out there in philosophy of biology for this is, Samir Okashi uses the phrase cross-level byproduct. You get group benefit without group selection. It's a byproduct of individual selection. And I use the phrase fortuitous group benefit, where you get, again, group benefit without group selection. Here's the thing that I wanted to ask a question of Silvia about. So she mentions Lewontin's 3 elements for evolution by natural selection. And she thinks that this picture needs to be replaced when collectives do not have established reproduction with this identification, recurrence, and genealogy. So one question I have here is, I'm not sure what these last 3 concepts mean. For example, if collectives don't reproduce, what does it mean for them to have a genealogy? So let's put that aside for a minute. But here's the from the further discussion in her talk, I understand this is what she's talking about when she says collectives lack established genealogy. It's this distinction, which is important in Ford's talk also. So on the left, you see groups reproducing, as it were, uniparentally. Each daughter group has exactly one group ancestor. It's like asexual organisms reproducing, budding off. There's this other way that groups can reproduce, which is they send out migrants who mix together and then they settle down and form daughter groups. That's an important distinction. So with budding off, group reproduction is uniparental. With global migrant pool, groups reproduction is variably parental. Each daughter group, as was said earlier, derives from between 1 and n parent groups. There's an issue here about how group fitness should be defined, but since that didn't really come up in the presentations, I'm not going to go into that now. I do want to talk about something that I took away from both Sylvia's talk and Ford's. Suppose daughter groups are formed by random sampling from a global migrant pool. We're looking at this right-hand model here. That's what we're doing. And the daughter population is just random samples of size n to establish the next generation of groups. Here's an additive model of how altruism and selfishness might work. And what I want to say is that if you're forming groups at random, and you have an additive model of fitness, you're not going to get group selection favoring altruism. Sylvia and I agree about this. This is an important point. So in this model, why is it additive? If your partner changes from selfish to altruistic, you gain a benefit B, regardless of whether you're altruistic or selfish. To move from right to left in a row, you're adding B in both rows. Similarly, if your partner is altruistic and you move from selfishness to altruistic, you pay a cost C. And the same is true if your partner is selfish and you move from selfish to altruistic. So you see how going cross rows and up and down in columns involves adding a constant. That's the simple, obvious meaning of why it's called an additive model. This is a model where the groups are of size 2. The point I'm making is true no matter what size the groups are. It's just easier to visualize. You can derive from this additive fitness arrangement a criterion for when altruism will be fitter than selfishness. And the criterion is the probability of your partner being an altruist given that you're an altruist, minus the probability that your partner is an altruist given that you're selfish, has got to be greater than the cost-benefit ratio. And when you have random formation of pairs, the left-hand side of that inequality is 0. That's what failure of correlation. They're independent of each other. And if C and B are both positive, you can't have altruism being fitter. You can't get altruism provoked by group selection. However, if you have a non-additive, an interactive model, you have this new constant I in the upper left-hand box, and it's a non-additive model now. And what I just said about the additive case is exactly wrong now. You can have groups formed at random, and it's perfectly possible that altruists are fitter than selfish individuals here because of that interaction term not being 0. So random formation of groups drawn from the global migrant pool doesn't spell death to the evolution of altruism. It does if you have an additive model of fitness. With an interactive model, you're fine. Although it's nice to have correlations between, people in the same group tend to be the same. That helps altruism to evolve, but it's not an essential feature here. Okay, Sylvia and I agree, I think, about pretty much everything except the thing about the Lewontin formula and its role. Let me turn now to Ford's comments. Okay, what is the too many parents problem? Ford says that biparental reproduction is small enough, 2 is a small enough number for evolution by natural selection to occur. But if each object has 20 parents, this may be too much, though there are no hard and fast rules. And Ford is interested in community selection where there's a global migrant pool. And he's skeptical about community selection in this case. In this case, just to make sure we're all on the same page, community selection, the word community means an ensemble, a multi-species ensemble of organisms that are affecting each other's fitnesses. So you and your microbiome is a multi-species community, which I'll get to that in a minute. Ford's point about 2 versus 20, it's nothing special. I mean, I think the thesis that he's endorsing, which Godfrey Smith endorses also, is that the number of parents is a factor here when you have group selection, when you have community selection, when you have any kind of evolution by natural selection. So I want to talk about the 2 versus 20 problem, where we're talking about group selection. And by group, I mean groups of conspecific organisms. There's no multi-species communities now. Okay, so group selection here, all these groups, the G's and the F's, are all conspecific organisms. Ford is right about this mathematical fact. Suppose you draw S samples, each containing B balls, randomly from an urn. The samples will differ from each other more the smaller B is. So drawing pairs of balls, B equals 2, is going to result in more variation between samples than if you draw B equals 20 balls. That's right. What is its significance, however? In terms of getting enough variation among samples, the number of balls in the urn needs to be considered here. Notice the previous slide, I didn't say anything about how many balls there are in the urn. I said you're going to draw a bunch of samples, each of a certain size. Are there 1 billion balls in the urn, or 50, or what? If the urn contains 10 to the 10th balls and you draw 20 balls for each sample, there's going to be plenty of variation among samples. So the idea is an absolute number here. I'm not sure exactly what it is. What's relevant is the size of the daughter populations that are created compared to this global migrant pool, which could be stupendously large. So I don't buy the too many parents argument. I think 2 and 20, fine with me. I mean, in terms of group selection, cool. So to summarize this point, there will be more variation among descendant groups if there's budding off than there will be with a global migrant pool. True that. But if the global migrant pool is cool compared to the size of each daughter group that's formed, daughter groups randomly sampled from that giant pool can differ a lot phenotypically in terms of their fitness. Group selection can do its thing. I want to mention the context of the footnote that Ford was discussing. It's in a brief section in which I'm talking about community selection. And the main example, which I talked about only briefly, as I said a few minutes ago, was the human holobiont. So here's what that is. Probably most of you know this. About 39 trillion bacteria, fungi, protozoa, and viruses live on or inside of each of us. Isn't that icky and weird? These microbes come from about 10,000 different species. Some help us by stimulating the immune system, degrading toxins, and synthesizing vitamins and amino acids, but others are harmful. A baby's microbiome mostly comes from its mother, and this 2019 study reports that after around 3 years, the relative proportions of microbial taxa remains mostly stable, but the microbiome's composition can be altered by diet and by taking antibiotics, of course. So what does the human microbiome look like? It's not that we and our microbiome come into this giant migrant pool and then there's random sampling. I mean, it's more like budding off from mom to offspring. It's not exactly like that because there are other things that are coming in there too. So I guess that's all. That's it. Thanks.
So, thank you everyone. Now it's the time for asking the questions that the room would like to ask to the audience and the commentator. Also, maybe the commentators want to signal something that maybe others did not understand, or maybe they disagree on something. That would be also interesting. We're going to keep that discussion to something very small because we know everybody is very tired. But anyone who is willing to make a comment or ask a question can do it now. And it's also true on Zoom. So we'll see if there's any question or comment. Who wants to start?
Yeah, so thank you again for your time.
Oh, I'm sorry, I forgot to say, if you could come here to ask a question, that would be great.
Oh, yeah.
Especially for the people on Zoom, because otherwise, Elliott, I think you can stay here. Just that if you're close enough to the microphone, I think it could help.
I didn't know this was involved in this. Okay. I guess now I have to do this. Now I really have to do this. Okay. Well, I guess, yeah, thank you. I see I just finished my PhD on the history of evolvability and evolvability as the greatest example of higher level selection. But I've encountered a lot of issues from evolutionary biologists, most prominently, you know, some of the best evolutionary biologists that we consider the highest status evolutionary biologists. Like I've talked extensively with Doug Futema about this. I grew up with Richard Lenski and yeah, many other. I'm writing a paper with some evolutionary ecologists right now, and Andrew Hendry, I don't know if you know him, but he also does not believe in higher level selection. So I guess my question is, how can we entice evolutionary biologists to engage with and believe in our work? Because I do believe that it's valuable, but I believe that we have an issue between science and philosophy where a lot of the scientists aren't reading our work and believe in what we do. So, yeah. Can I be done? Can I go sit down, or do I have to be up here? Okay. Yeah, this is really weird.
So, am I right that what you mean by evolvability is a property of the species to avoid going extinct if it need be evolving new characteristics that'll help it to cope with new environments. That's in the neighborhood of what you mean by that?
Yeah, I mean, there are 25 different definitions available in literature, but that's, yeah, very general terms. What I focus on genetic evolvability, precisely like how sex and mutation mechanisms actually allow some species to outcompete others. And a lot of nuances in that. But yes. So it sounds like the opposition you're hearing is not about worrying about evolvability. It's about bringing in group selection to explain evolvability. Is that right? Or what I found is that a lot of these researchers find higher level selection as a theoretical possibility, but they don't see a whole lot of empirical evidence for it, or like the empirical adequacy of science isn't there to support this idea. I guess how can we convince them otherwise?
Well, I mean, I think they're good examples, but they quickly lead to sort of terminological issues. So I think the evolution of reduced virulence in viruses is a great example. Some of that is group selection. Sex ratio evolution, another great example. And as a philosopher, you don't get surprised when the tangle that you get into often ends up with people using different definitions of what group selection means. They'll think that a certain argument shows that all selection is strictly individual or genic selection. Then you have to think about what that all means. So there's a lot of conceptual clarification that needs to be done.
So Ford Doolittle has raised his hand. Can you speak?
Yes. Yeah. Can I speak? Let's see. That's apparently I can speak. And I apologize for my inability to speak when I was giving my talk because I think I was garbled. But anyway, I think Elliott he talked about holobionts in some way, and I'm not sure, and I don't think anybody is sure how many of the strains that you get from your mother you actually pass on to your children. Which is the irrelevant issue, right? I mean, it doesn't matter how many you get from your mother. If you exchange all of those from your diet or your pets or whatever it is during the course of your lifetime before you reproduce, then it doesn't matter. So, and I don't think that we know that answer yet. I mean, there are data, but they're not particularly well defined.
I agree. I mean, yeah, that's an empirical question. That's important.
Yeah.
Yeah.
That was my comment.
Thank you, Ford.
Can I stand here?
Oh, should be okay.
Should be okay. One more step. Okay. I want to thank you for your insightful talk. I'm coming from a biologist perspective, so I have two questions. I think both of them relate to the question of choice. In the situation where you have the honeybee, the beehive, the worker bees that are sterile don't necessarily have a choice because it's the pheromones that is released by the queen bee that actually dictates whether they are fertile or not. And the question of the genetic code, we also don't know if any other genetic code can actually be viable on Earth. So there's also a question of choice. So it seems like most of what we're discussing here is an aftermath of not having a choice of being in this religion. So could you maybe expand?
Sure. The argument that Crick made it in the 1960s about the universality, what he thought was the universality of the genetic code being a killer argument for universal common ancestry, it requires that there's got to be more than one workable code. Otherwise, it's not an argument at all. And so I think he assumed that, of course, with very limited. I mean, it was sort of a plausibility argument. There's got to be one. So my impression is that in the last 20, 30 years, there's been work on the variability of the sort of workability that different codes would have. And there are even some papers that argue that the code that we mostly have is optimal, according to some interesting criterion.
So my picture, I mean, I haven't really kept up with this literature, is that the kind of received wisdom is that other codes are possible. And so that satisfies the condition that that's what it would take for something like that to be evidence for common ancestry. The first thing was about the honeybees not having a choice. Yeah, I don't think you need to have a choice to be an altruist. I mean, you're just born that way and you can't help it.
No, so that's the thing, right? It's the selfishness of the queen bee that dictates the altruism of the group. So when you discuss group dynamics of group evolution in that characteristic, you still require one participant to be selfish in that nature. So I fail to see how you're involved in that.
So let me change the example from, I guess this is not honeybees now. Let's talk about nests that are founded by more than one fertilized female. And I'm going to go to sterile workers now, although it probably wouldn't matter. So you can imagine, or yeah, it'll be the same either way. You can imagine there are two types of fertilized female. One of them produces offspring, some of which have barbed stingers, others do not. The second type produces only no barb stingers. And suppose they're fertile, I mean, if you like. So group selection will favor the ones with barbed stingers. Within group selection will favor the strategy of being a free rider and not creating barbed stingers in your offspring. Let somebody else commit suicide. You see?
Oh, sorry.
So I just want to say, I just would really like to hear. So I really like the kind of model that Kevin was putting forward, which I first got from Alex Pradeu's work, and I think there's lots of other examples of that, right? I don't see it having anything. I mean, it should be about the direction of phenotypic variation that's chucked up, not about directed mutation. So I'd really like just put directed mutation to one side and instead talk about how developmentally induced correlations between multiple phenotypes might bias the direction of phenotypic variation. I think that's much.
So it's about development. It's not about mutation. Is that your
exactly? Well, and it's about how the developmentally induced correlations between multiple phenotypes might mean that when you're trying to select for an optimal trait value, you're getting a package which reflects the capacity. I mean, let's say Alex's work was all on. Yeah, there's lots of other examples. Anyway, so if we could just, I would really like to get you two talk about the issue, which is really interesting, without it being about directed mutation.
Why don't you go ahead?
Yeah, for me, it's not about guided. It's not about directed mutation.
Oh,
it's about how the history of selection can influence the phenotypic consequences of mutation. So, for instance, if we were thinking in terms of a thought experiment, your bear one is not the right thought experiment as far as I'm concerned. Actually, you have an example in your new book, which is kind of closer, which we could use, where you think about what happens if we've got two strains of bacteria, one red, one blue, and different color backgrounds. The point is that if you impose disruptive selection on that kind of system, and you're selecting on color, so there's a history of selecting first for red, then blue, then red, then blue, then you end up with a population that's very good at switching from red to blue, right? And conversely, if you impose a different form of selection on that system where you're imposing stabilizing selection, you're only always selecting for red, then they'll be very bad at producing blue mutation. And the reason that occurs is because selection is favoring different forms of epistasis and pleiotropy. And the net consequence of that is the shift of distribution of phenotypic variants to produce variants which are facilitated in the sense that they are more probable as a result of that historical selection regime.
I misinterpreted you. I withdraw that. I'm really glad, because I thought I was going to have to sort of learn a whole lot of new stuff, and you know how hard that is. So.
Also, that's not my thesis. Those are
No, good. I'm happy twice over.
Other questions or comments?
Et voilà.
Other questions? Critiques? It's a fantastic opportunity to discuss with these great people. No? What do you say? Oh, yes, he's here.
Nice to meet you again. Not my field, so I wouldn't ask except everybody else is bowing out. But thinking about the multiple parents problem, sounds to me very much like a graph modeling problem where you have a number of nodes, they have some connectedness to each other, to the next generation, so maybe a graph modeling time series. Are you aware of sort of work that's in that field of saying, given some arbitrarily complex community, you know, whether it's within a species or not, you know, modeling probabilistically, what are the kinds of or level of inheritance that one snapshot should have with the previous time point or the previous time point?
No, I am not.
Oh, really?
So the
That's surprising.
I mean, well, it's just so my entree into this was thinking about the global migrant pool and how that works, but as sort of the more general mathematical framework, no, I don't really know about that.
Okay. Opportunity. Thank you.
Sounds like a good discussion topic for dinner later, but any other question, critique, comment? Please.
I want to keep asking, but I have a million questions. So I'll talk about it later on. But so you talked about, and I know you mentioned this in nature, so that you conceive of fitness as adaptive. So do you believe that there is no fitness going on when the environment changes and then population less adapted to existing circumstances? Oh, sorry. Do I have to come up and say that?
No.
Okay.
So when the environment changes, traits will change their fitnesses. You know, maybe that is having, you know, thin fur used to be okay, now it's not so good. And I'll think of that as a change in fitness. Let's just think about it as the probability of surviving to adulthood. So the contrast that I think is really important is between whether this bear or this trait survives, increases in frequency. That's an observation. To say that happened because there was a fitness difference is a theory. And you can get change in trait frequencies without fitness differences. And you can have fitness differences without getting change in trait frequencies. So these are really different things. So to me, the explanatory concept is the ability concept, the propensity concept, what I call fitness. And yeah, but there are, you know, practitioners who want to back up and think about what I'm calling the observations, namely the birth and death rates of traits as a kind of, that's the machinery for a theory.
Okay, I think it's time to really thank Elliott and Kevin and Silvia and Ford for a fantastic afternoon. So thank you very much.